Who's ready to see some cool stuff? Let's see if this works. Hello. All right. Here we go. I'm going to try to do something a little different today. Not a video game. Plankton. So, this is going to be a little different for me because I've actually never sorted plankton quite this way. Normally I'm looking through a microscope eyepiece. And, right now I'm looking at a microscope screen, or a computer screen. It's kind of different. It's going to take me a hot second to get my feel. Alright, what are we looking at though? These are plankton samples that were collected off of Hawaii in 1971. They went into storage. Nobody's really touched them in all that time. And so that's what I'm doing now, is going through those samples and pulling out the fish larvae that have been in those samples, right? We're going to add those to the museum collection. Yeah, Daisy, you probably don't want to be sitting up here on this one, baby. Kitty Cat's a little worried about what's going on right now. Okay, so again, these were samples that were plankton samples. So these samples have all sorts of stuff in here. We're primarily interested in the fish. That's what we want to take out and save for identification later on. And so I have a jar here that has just the fish that have been pulled out from these samples. So that's what's in this jar here. Just rolling around. There's a lot of fish in here. And we're going to get more. The plankton sample that we're processing right now is about a gallon. So there's a lot of material in here, and we're going to just go through it and pull out the stuff. What we're looking at now is actually the head of a stomatopod. So that guy, that whole little dude there, is not a fish larvae. That is a stomatopod larvae, like a commonly called a mantis shrimp. We got those. All right. This sample we have found has a bunch of these in it. These are the fish that we're after. Well, we're after all fish, but we're after these in particular. Like this sample tends to have a bunch of these in it. Focus right. Down. There we go. This is what we're looking for. So this is a fish. This one is in the family Molidae. So we're just going to be finding these little dudes and pulling them out. There he goes. Now he's in a little jar. Since this is the raw plankton sample, we're just gonna pull out the larval fishes and separate them from all the other stuff that's in here. You see there's one here. Oh my gosh. And all this big like mass of other stuff. We got some shrimps in here. And then we got, you see there's a couple of little more molids in here. We need to get those out of all the junk. Just want the fish. We don't want all the other stuff that's stuck to them. And mollas tend to be very spiky. They have these dermal spines that make means that everything sticks to them. There we go. So I have this guy free. Come with us. Another one. Little fishy. Hey, Featherburb. Thank you for that follow. I appreciate that. So you can see there's a bunch of like shrimp in here as well, and they're all in this like gelatinous matrix. And occasionally I see like this thing here. We've seen a lot of stomatopods in here too. These mantis shrimps. So there's a bunch of those in the samples. So these are going to be all tangled up inside, I think, with all this big pile of goodies. Like there is. All right, so there's a little molid. So again, this is a molidae. We would know those commonly as the ocean sunfish. Ocean sunfishes are the, the weird ones that lack the tail that can grow to like 2,000 pounds. We know them commonly as like mola mola, right? But this one, there are actually quite a few species of those, and I don't actually know if this is mola mola or if it's one of the other species. So typically what we do when we're doing this is we just sort the samples. We just want to get the fish larvae out, and then we'll go back through once we have all the fish larvae separated out from all of the other zooplankton and do another run over the taxonomy and make sure that we get all of the, get the proper identifications on things. Yeah. So at this point, it's all about just recovering the fish. So this is a mola. Again, they have these little like dermal spines on here. They actually have like names for the different spines and the positions of the spines. I'm trying to orient it. That's the tail, little tiny tail down here, little mouth up here. So very, very strange fish. And again, they grow to 2,000 pounds. So it's not as fish. This is a crab zoea. Check out this rostrum on this thing. It's huge. So I think these are porcelain crabs. So we're gonna find lots of crabs in here and lots of shrimp in here as well. Mola. Oh, that's that's the tail of a stomatopod larvae that's fallen off. It's the claw of a stomatopod. I don't really know much about these samples. I don't have all the data associated with these, so I don't know. I know where they were collected and I know when they were collected, but I don't know what type of gear they were using, and I don't know how much volume of water was sampled. So we're gonna check out this thing. There's the crab body, and then look at the rostrum. Crab body. And the rostrum just keeps going. It's huge. There's another fish sitting here next to it. This little guy is another fish. This one's called a gimpylid. A little bit scuffed up, but I can tell it's a gimpylid. These are commonly called snake mackerels, but I can tell based on the dorsal fin and the anal fin based how spiny they are. It also has spines on its head. Gimpylid in there. Sometimes when they get clumped up like this, you gotta move things around. Another little molid there. So many. We've been finding tons. This sample has just been loaded with molids and the stomatopods and these shrimp. It's basically what this sample has amounted to, with a couple of other random things mixed in. That is just going to be some trash. That's interesting. So this is my first time, my first day trying to do a plankton sample like this on stream. We're just getting it all set up. So we're learning a few things here. Like the camera's not set up perfectly. It looks a little bit off to the side. It's also got stronger magnification than the regular lens. So it should be zoomed out a little bit further, which we're zoomed in really tight right now. So I think the camera is adding some artificial magnification, which makes it a little bit trickier to navigate and orient. We'll figure it out. Maybe there's a setting somewhere that I can adjust the magnification of the actual camera. It'd help if I did this. Try to orient it a little bit better. There we go. Look at that guy. Is that not a cool fish larvae? Yeah, so again, a mola. Family's molidae, oceansunfish. You know, it's got these dermal spines that are off of it. Weird little tail, tiny little mouth. It's a real good view of it. Molidae are they have the highest fecundity of all fishes. So fecundity is a measure of how many eggs a fish can produce at a time. And so molids are like the masters of this. They have the highest fecundity of all fishes. They can produce, a female can produce 3 million fish eggs at a time. So if you think about that, 3 million fish eggs is an insane number of eggs to produce at a single time. Because ecologically speaking, right, if we just think in terms of pure ecology, macroecology kind of stuff, all that a female needs to do over her entire lifetime is produce 2 offspring that live to adulthood. She's got to essentially replace herself in the population, and she's got to replace a male in the population. So as long as she has 2, a male and a female, that replace in the population, that survive to adulthood, then there's population stability. If she produces more than that, if she produces like 3 that make it to adulthood, then what happens is we have population growth. So she's producing more offspring than are dying out. And so each time she spawns, she releases 3 million eggs. So, like, if you had even 99% survival of that, the oceans would be just full of mola mola. So this is actually one reason why I like working with larval fishes, is thinking about the natural mortality that these fish are naturally going to experience anyway. For me to recover some of them and study them doesn't feel as bad as if I recover an adult. If I go out and collect an adult who has managed to survive to adulthood and kind of run through the gauntlet of life to make it to adulthood, I feel a little bit worse about collecting them. Larvae are way more abundant in the wild. Eyes of that stomatopod. Crazy. I'm not an invertebrate biologist, so I don't know what these shrimps are. Some kind of shrimp. Loads of them. I think another thing I'm going to manage to do, again, first time streaming like this, we're going to change a little bit of the setup. I'm going to swap the light around. So right now, I'm often covering up the light as we're moving the sample around. Yeah, this sample is wild. I don't see hardly any copepods. I don't see very many chaetognathes. It's basically just shrimp, stomatopod, molids, and occasionally something else. Oh yeah, these porcelain crabs. Again, some of these are getting really tangled up in the other zooplankton, so see one mola sitting there now. I think I'm trying to get out another one right here. Where are you? This is really entangled. Try to clear it off as best we can using the forceps without damaging the specimen itself. There we go. We got it mostly free. Alright, I think that's mostly it for this tray. It's a weird little gastropod right there. Some sort of mollusky thing. See that little... See the little spiral shell, and the dark spot's the eye spot. Pretty neat. Alright. So that one tray is done. It's time to go get another one. Be right back. Daisy, do you have any idea what's going on? All right, so I am like processing this in my kitchen sink, which is a little bit mm, interesting. I did a lot of pre-processing of these samples before I brought them home, so that they are safe to handle in my kitchen. All right. Again, just tiny little dudes, right? So these were sampled from the Townsend Cromwell, which was a U.S. Fish and Wildlife Service research vessel in 1971. The Townsend Cromwell operated off of the Hawaiian Islands. It was based out of Honolulu, I think it was. So they operated off of the Hawaiian Islands for many decades. These samples came to us from some cruises that were done in the late 1960s and early 1970s. So this one was done in 1971. So it always makes me feel a little bad that all this effort goes into collecting these samples and then they sit on a shelf for 50 years without anybody taking the time to do anything with them, you know? So part of what I wanted to do was go through these, sort them out, pull out the fish larvae, add those to the museum collections, and then hand the other zooplankton off to our crustacea group so that they can have them. And they can do whatever they want with them. They can leave them on the shelf for another 50 years, I guess, or they could figure out what these shrimps are. Yeah, it just feels sad when things are collected and then not ever used. Kind of a waste. And yeah, nothing but shrimps and stomatopods in here. I know there'll be some other stuff that pops up randomly, but we did find the compiled larvae, snake mackerel larvae, but I don't know what else we're going to find. So here's a good look at a stomatopod larvae. So stomatopods are commonly known as they call the mantis shrimps. So you can kind of see one of the things they're really known for is using their arms to be weapons. So that's what that little guy is there. Got a real sharp point to it. And they use it to lash out and spear prey. or to stun prey. So they kind of like a mantis, praying mantis, right? They look kind of like a praying mantis, and they function like an underwater praying mantis. Pretty fun. So you notice there's like different sizes of them here. That's a small one, but we have, I mean, here's, this one is a giant one. Look at that. Tail starts there. And then there are the eyes. This one's huge. This one's almost, I would wager, almost ready to transition to an adult. Hang on, let's see if I can fix this light. The light's kind of annoying me. Getting in my way. Okay, now I'll just move the light. So I won't be blocking it now. I do want to get cable out of the way. There we go. Okay. Again, first day trying this. I don't know if this is going to work or not. Going to be a lot of little bugs to work out. Everything from camera setup to audio to my desktop, my working space. I'm actually not seeing any fish in this one. Just a couple of mola, and then another one of those porcelain crab things. I think those are porcelain crab. Let's see. I have a gigantic pile of books over in the corner over here to help me with some of these IDs. Again, we're not going to identify this thing, but just at a cursory level, I want to know. I know it's some sort of crab larvae, but I don't know what kind of crab that is. So, let's see if we can get an idea. Crab larvae are like seriously difficult to identify because they go through multiple stages. Crabs can go through like as many as 8 stages in their development. It's just wild. Let's have the big rostrums. Well, this might also be a pea crab. I don't know, lots of different types of crabs out there. Mole crabs and sand crabs. Anyway, lots of crabs out there, and that's one of them. Hopefully, like, a decapod person can figure out exactly what that is. I'm not a decapod person. There's a completely different type of shrimp right there. Look at that guy. I don't know what that one is. Different from what we've normally been seeing. Wild. You know, there's so much in these samples to like figure out taxonomically if you wanted to identify all the stuff that was in a plankton sample. It'd be so hard to do that. It definitely takes specialists and specialization in different fields to really get a handle on these things. All right, that one's done. Only a couple of molas in that one. Alrighty. Fresh tray. Again, giant stomatopods in here. Giant mantis shrimp. Not giants, but these are not too far from transitioning to adult mantis shrimps and settling down on our reef. Those are pretty big and advanced larval stages of the mantis shrimps. Let's see. Ichthyoplankton, which can often be confused for fish. Hey, Yoda Main, how goes it? These little dudes often trip us up when we're looking for fish because they have sort of this long body. They also have a very different type of head there. How's your weekend going? Yeah, this sample is full of stomatopods, mantis shrimp larvae, and shrimp. And then occasionally we're finding some molids. Yeah. Yeah, true. That's going fine. Did some stuff trying to get this set up mostly. This is definitely the highlight of my day. Not too many fish. There's one finally. Look at that dude. So this sample has had a bunch of molids in it. So there's another one. Good look at that guy. You know, typically when we sort plankton samples, you know, I was asked a question about how many do we want out of a sample, and it kind of depends on. It depends. The answer is it depends. So if you're working in a consulting firm, which is how I got my start doing larval fish stuff, time is of the essence. Every minute that you spend working on a sample is being billed to a client, right? So they want you to move fast. So we actually put a cap on the organisms that we put in, or that we would pull out. So we would only sort out up to around 200 individuals, 200 larvae. And then we would stop and then just sort of extrapolate depending on how many times. So we have a device in the lab called a plankton splitter, which you basically does what it says. It splits the sample in half, and then you could split it in half again, and you could split it in half again, and eventually you're working on, you know, 1/2 of the sample, 1/4 of the sample, 1/8 of the sample, 1/16 of the sample, like that. And then that'll save you a lot of time. And then you just multiply the numbers that you get based on how many times you've split the sample. Saves you time and gives you a pretty accurate estimate of, you know, sort of broadly speaking, what's in those samples. But look at the eyes of the stomatopod, crazy. But now I'm at a museum, and so we actually want the rare stuff. We want the weird stuff, which means we're going to process the entire sample because there might only be one rare larvae in this entire sample. One larvae that's in like exceptional condition that we would want to use if we're teaching a workshop, or if we're talking about, you know, if we're doing a study on one. We want one that's like in like the perfect condition. We'll spend the time now to find the one good study organism. So that's what I'm doing now, is I'm going to go through these samples and pick out every fish larvae I can find, because I'm hoping that some of them are going to be rare, some of them are going to be uncommon, or some of them are going to be in very, very good condition. So like in the consulting world, you wouldn't want to spend 20 hours processing one sample. That would be a lot of time. But in the science, like the museum world, spending 20 hours on a sample means you're likely to get something good out of it. So sure, spend that time. Anyway, let's go get a fresh petri dish. So when I talk about processing the whole sample, I just talk a little bit more about what that actually means. So this is the sample that was collected in the field. It's put in a gallon jar. Yeah. So this is the sample that we're working through. And then, we want to recover all the fish larvae that are in this particular jar. And we do that by going petri dish by petri dish. So I'm pulling out petri dishes of sample at a time. So again, it takes time. We're 45 minutes into the stream, and I've done, 4 of these maybe? So it takes a long time to go through an individual petri dish, and you think about how many petri dishes worth of sample there are in this giant 1-gallon jar. There's going to be a lot of material to process. And again, when you're working in a consulting firm world and you're charging a client by the hour, they're not going to be happy when you're spending that much time getting that kind of data. So you gotta manage the time a little bit better. I don't care. I'm doing this on the weekend for fun, so I'm gonna get everything that's in here. And then we'll hand the samples off to our. So there's a standard time that's expected. Not really, but in terms of like budgeting, right? When you do a contract with a when you do a contract, there's a certain expectation of what that budget's going to look like, and so you have to plan ahead with the number of hours that you're going to expect to process that sample. So, is it going to take you? Oh, we're going to go out and collect 20 samples a month. How many hours is it going to take to process 20 samples? And that's what you need to bill the client, right? If you say I'm going to go collect 20 samples a month, and it's going to take me 100 hours to process each sample, that could be a pretty hefty bill that you're handing to the client, and the client might not be happy about that. On the other hand, if you tell them, oh, we can do 20 samples, we can do that in 40 hours, that's a week's worth of work, and then it turns out to take 100 hours, as a company, you've kind of hurt yourself pretty bad, right? Financially. So you've overestimated the time it takes to process those. So you're paying your own people more than what you're charging the client. So the consulting world's like a balance, right? That's why they always overcharge. I know I was, when I was working for the consulting firm, I mean, this was again 20 years ago, I was getting paid like 20 hours or $20 an hour was basically my rate that I was being paid. But those hours were charged at $60 an hour to the client. So they charged the client a lot more for my time than they actually paid me. It's like, dang. Why is that? But part of it was because in case I had to work longer than expected to finish up a job. And also that extra money is how they fund, how they keep the lights on, right? How they pay the power bill, how they have administrative staff who don't do work that brings in clients. So they can't bill those hours. Like you can't charge a secretary's hours to a client directly, right? You got to factor that into all of the grants or all of the projects that you're putting in. I managed to get the projects that I was working on finished in time and under budget, so I got a nice bonus at Christmas. That was my incentive to work well, work efficiently. Was if I did my job really well, they would pay me. I got like an extra paycheck around Christmas time. Ooh, there's a different one. So we've been looking at stomatopod larvae, the mantis shrimp larvae. This is a lobster larvae. Dude there. So with a round, I don't know what these things are. I don't know the anatomy of these things, but these are like round with the legs sticking off, a little bit smaller. So that's the lobster. Compare that to the stomatopod that has sort of the rectangular body to it. So it's pretty neat. So that's the stomatopod there. That is the lobster. Way too many legs. I think they have more legs than they need. And yeah, I don't know what these shrimps are. The shrimps are shrimps. Shrimp on top of shrimp on top of shrimp in here. So this one is like, this is a mantis shrimp that's almost ready to settle out. You can tell because it's already getting like, instead of being transparent, it's starting to fill in, right? So that thing right there, that's about to settle on a reef. I think we lost its arms. Yeah, its arms have fallen off, but this one's like barely plankton anymore. We just happened to catch it in a plankton net. There's a fish. This little guy. There's a fish. All rolled up into a ball. That's its eye. Not a very good example of one of these, but this is a flatfish. This is a bothidae, a left eye flounder. But it's normally they're like flat and look like, hi Daisy. They look like almost like feathers or leaves, and this one's just like curled up on itself so that it's hard to see. But I know there are some others in this sample. I'm hoping that we'll find another one and I can show you what a proper flatfish larvae looks like. That was one, but not a very clean example of it. Here's a fun one. We haven't seen one of these yet either. This is a type of shrimp. This one. So that is a type of shrimp. It's missing one eye. This thing's, the family of this shrimp is Luciferidae. And there are two genera of Luciferidae. One is Lucifer, and the other is Beelzebub. So That's about all I know on those shrimp. It's just that there's some sort of devil shrimp named after Lucifer and Beelzebub. We actually have a paper in the works on those that talks a little bit about their ecology in the Arabian Gulf. It got reviewed and got rejected with some revisions. So I've got to make some revisions and send it back in. But the only reason that I really secretly, just between you and me, the only reason that I wanted to publish on that genus of shrimp or that type of shrimp was because I wanted a paper talking about Lucifer and Beelzebub. Like I decided to do a project on them just because of the name. That's all that's in here. Lots and lots of shrimps. All right. Let's go get more fish. Chat, it seems the cat has stolen my chair while I was away. Okay, I gotta go. Come on. Out of the chair. I know. What if I gave you a cat treat? What if I bribed you out of my chair? Oh, that works. Come get a cat treat, Daisy. Yeah. Alrighty. She was just keeping my seat warm for me while I was away. All right. Yeah, I've never used this microscope before, so I'm still trying to get my bearings on where things are. It's the mola. It would help maybe if I did the light. Okay. So yeah, I've worked for the consulting firm for 3 years doing this job. From 2005 to 2008 when I went back to graduate school. And I listened to a lot of music and a lot of audiobooks. And I would have listened to a bunch of podcasts, but podcasts weren't a thing back in 2005. So I listened to books on CD that I checked out from my local library. A lot of a lot of David Baldacci novels, I remember. Spines on these little shrimp or crab larvae. I don't know how these things function. It's like things sticking so far out in front. I don't know what they're doing to those things. Shrimps. That, I think. Fish. So this is a gastropod that lacks a shell. That little thing right there. So it's a mollusk that's free swimming. Just kind of swims around, flaps around. Doesn't really have much of a shell. There's another mola. This sample has just a gazillion of these things. Because we have limited data from these cruises, the cruise where these samples were collected, I basically know where they were and when they were there. I don't have any information about the gear type they used or how much, like, the volume of water. Well, the key thing is I don't know how much volume of water they sampled, which means I can't calculate densities. All that we can do right now is just do like presence or absence data, the more valuable data is being able to calculate densities. But since we don't have all the data associated with these samples, that's a little bit problematic. So kind of all we'll be able to do is just pull out the specimens and use them for like species-specific research, less ecology research. Look at all the spines on this guy. We also only wound up with like a random collection of samples from these cruises anyway. We don't have a full cruise worth of samples. So it's very kind of scattershot. These samples we know were collected in 1971 around Hawaii, and we have like the rough GPS coordinates of where they were collected. So that's helpful, but you know, normally we want more information about the volume of water that was filtered per sample, so we can calculate density. We want information about water temperature, salinity, chlorophyll concentration, oxygen, you know, those sorts of environmental parameters that influence biology. That lets you do more ecologically focused studies, and that information for these samples is lacking. So it's a little bit of a bummer, but it's also not the end of the world because there are cool samples, or there are cool fish in these samples. So not terrible, but it could be better. So I already have decided that I need to get a little side table, because right now I'm getting up and running over to the sink every few minutes to sort of swap out these samples and get fresh material, and having to run behind my green screen to get into the kitchen. The whole reason I have a green screen is so that it blocks off my gross, dirty kitchen from from my viewers. But right now I have to run around it. So I think in the future I need a little side table sitting over here where I can process the samples a little bit faster. Also, I won't disappear for a few minutes. My plan, in the grand scheme of things, is to start doing these on like, I'm going to start doing these on Saturday, these plankton sorting streams, making them more of a regular thing. Do these on Saturdays, and then pull out little videos about the stuff that we find on Sunday and get those uploaded onto the YouTube channel. Because it turns out there's not a lot of information on YouTube about larval fish taxonomy. That's something I can do that's a service to the community. Oh man, that one's totally tangled up in there. So this poor fish is like completely tangled up in just gunk. Some sort of gelatinous organism thing that got wrapped around it. And then I wound up using the forceps to gently sort of peel it out of there. Hopefully without damaging the specimen. And get most of that stuff off, that's good. I got this petri dish a little fuller than I would like. So when that happens, when there's too much stuff, it's best to like pull it all off to one side and clear out a little space in the petri dish and sort of just work through the cleaner area. The danger is that the risk is that some, like a giant pile of shrimp in here, they might be covering up some of the fish that we want to pull out. So move things around, give yourself a little space. Seen any fish in a while that haven't been mollas. It's one of those crabs. Giant rostrum thing that looks like it's broken off. Stomatopods and shrimp. Well, it's just an eye. Some sort of invertebrate. Lost. Here's a mola. Like the fact that something like this is swimming around out there in the world just blows my mind, you know? I look at something like this and I just wonder, how? How does this thing function? What does it do in the world? It's got all these spines all over it. It's round. It's super tiny. I mean, you could fit like 5 of these on your fingernail. They have all these spikes that are all over the place. I don't know what this thing is doing. I don't know how it's doing anything. It must just like float. It can't do anything much more than just float around. eat the things that accidentally bump into it. I don't understand what these things even do. These things are so small that at thank you very much. So yeah, like I don't know how these things function. Let's go do this one now. Yeah, so when we have something that looks like this, like I don't understand how it functions in the world. There's another thing that happens for larval fishes that's, or just small things in the world in general. It's called, these fish live at what we call low Reynolds numbers. So a Reynolds number is basically a measure of the viscosity of a fluid. So low Reynolds numbers mean it's more viscous and that it's like stickier. So think of like the difference between pouring water out of a bottle and pouring molasses out of a bottle, right? The molasses pours slower. It functions differently. It's very sticky. So functionally, larvae of this size are swimming around in a fluid that is more akin to molasses than it is water. And I don't understand how they're able to do it. I mean, you can see on these things, it's got fins, so it's sort of developed, right? Let's see here. So you can see there's little anal fins sticking out there, right there. And there's a caudal fin there. So it's got some fins. It's able to move a little bit, but I just don't see how. It just blows my mind. Blows my mind. So I'd love to get like a good, like a 3D scan of them. Put them in a flume and like model the water flow around them. Again, okay, so this is one of those little gastropods. Sort of a shellless, or maybe there's like a little thin shell on this thing. Stuff to my forceps now. That little thing. So this is a little gastropod. There's a little really, really thin shell there. Pile of shrimp. So many. All right. That's a little one. So we've had a size range of these things in here, right? This one's like smaller. This might be one of the smaller ones that we've seen. there's a whole size range. You know, odds are both of these fish were sort of spawned and hatched at the same time. But one's feeding and the other's not, right? So this one has like much slower growth than the other one. That would be my prediction. We would have to age these larvae to figure that out. And to my knowledge, I don't know if anybody's ever been able to age larval mola. It's a very difficult thing to do. Shrimps. Some of those little gastropod things with the eye spots on it. I might be able to. I think those are in the guide here. Let's see if they are. Mollusks. Yeah, what mollusks are they? Yeah, again, I don't have a very good guide for the invertebrate zooplankton. I mostly have guides for the fish, the larval fishes. And I have a whole stack of those books over here, so a lot of times when we're seeing some of the weird zooplankton, I'm only gonna be able to guess at what that stuff is. When I was running a zooplankton ecology lab, I had technicians who knew how to identify copepods and all the other invertebrates in the samples, so I never really learned that many of them. I learned the main ones that we were dealing with in the Arabian Gulf. I didn't learn... and I learned them at like broad strokes. So Wad was able to take it down to the species level on most of the copepods. Fiji was really good at it too. They had done their master's theses on like copepod ecology and so they had spent the time learning the taxonomy of all the little invertebrate things. It's like over here in the corner. Okay. I think it's going to do it for this sample too. Yeah, I got too much in this one. There's a bunch of stuff stacked on itself. Hard to really see. Hard to pick out individual organisms when they're all stacked on top of one another and wadded up in a ball. Alright, be right back. my chair. Chat, the cat stole my chair. All right, kitty, let's get up. I'm ready to sit down. There you go. Aw, poor kitty. She got evicted. Alrighty. Bum, bum, bum. I did it again. I got a bunch of stuff in the sample again. Mollas. There are many species of mollas, so I don't actually know which one this one is. Just that it's molidae. I think some of the key differences are that some of them are round and some of them are a bit more like oblong, I guess you'd say. Oh. And that's a big pile of nothing. That's a ketognath. I'd expect to see a bunch of these. Oftentimes these get confused with larval fishes because they have like a really long body that can resemble some families of fishes, like Lupeids and Angrulids and menhaden and anchovies, right? But they are different. They're called bristlemouths. So they have these at the head end. They have these little chitinous hooks around a mouth they use to entrap prey, and then their body is like this long flimsy thing. They're one of the main predators, actually, in the marine environment. They eat copepods and fish eggs and fish larvae and crab larvae, and like they're not very discriminating on what they capture and what they will eat. Hold on. So again, what we're doing here is what we would consider to be a rough sort, where the goal is to just go through and pick out all the fish larvae, and then after we have all the fish larvae, you know, this little jar here is filling up. This is 70% ethanol and all the fish larvae that we've pulled out. Once we get that jar, not once we get the jar filled, but once we get the sample completely sorted, then we'll go back through and we'll look at the individual larvae in more detail and go through the books closer and figure out exactly what species they are, if we can. Like we'll do the it's called lowest possible identification level, LPIL. So we'll identify them as best we can, basically, is what that means. But that's far easier to do when we have the fish all out of this than it is to do it while it's all in this big pile of other stuff. Here's a fun one. It's our first eel. That is an eel larvae. That little dude. So all eels have a particular type of larvae that's called a leptocephalus larvae. So that's one of them there. Very long, very, very thin. This one's a little bit scuffed. It's been damaged a little bit, but that's be hard to straighten it out. But they're super thin. They're paper thin. They're so thin that they for a long time they thought that they got all of their nutrients just directly from the seawater, that they didn't actually need to feed, because they had never found leptocephalus larvae. It took a long time to find leptocephalus larvae with food in their guts. So the theory was that leptocephalus larvae, they're so thin that they must just absorb all the nutrients that they need directly from the seawater. But it turns out that's probably not true. Instead, what they eat is just marine snow, which is like junk, more or less. Marine snow is when you get sort of dead tissue or bacteria that sort of formulate into a little ball, and then eventually it reaches a certain density that's denser than the water, and so it starts to sink. So it sinks, and the leptocephalus apparently will feed on those little clusters and gobs of just dead gunk floating down. They've done some, I think it's been genetics work on leptocephalus guts. So like the gut microbiome, and it's all been this marine snow. So they are feeding, but they're just not, they're feeding on really poor quality junk. Shrimps and crab larvae. Just stuff piled up right there. So many stomatopods. My guess is that the sample must have been collected at night. And all the stuff that's in here, this all seems like things that come up from the bottom at night. Things that we would call diel vertical migrators. Have you ever heard that term? Daily vertical migrators, things that live down deep during the day, and then when things get dark, they move up towards the surface. We got two molas here that have a bunch of just gunk stuck on them. Try to use the forceps to gently peel that stuff away. They're not very clean, but that's fine. We got them out. So talking about how fierce the ketognaths are. I think this is a ketognath that has something in its stomach. Let's see if we can figure out what's going on here. So that's the ketognath body. So this is the ketognath's head. In a position where we can see the little teeth. If we go down the body, we can see there's a pair of eyeballs in there in the body. So it's definitely fed on something. Another one that's done the same thing. So the ketognaths are out here taking advantage of all these shrimps that are around. Yeah, so again, about all I know about these samples is they were collected off of Hawaii, which if you know much about Hawaii, there's a lot of reefs around, which might explain why all these mantis shrimp larvae are around. Mantis shrimps are a major component of reef communities. Another molid larvae. I'm expecting at some point for us to find some squid or octopods or something like that too. Seems like an area where we would find them. I haven't seen them yet. Generally going to be rare, so there might be a handful in this entire sample. Just haven't seen them yet, you know? This is some sort of crab thing. It's missing its legs. It's just the head part of the crab larvae. Wild. All right, I think that about covers that one. Let's go get another. Alrighty. So the past two, I've gotten a bunch of stuff in there, and so this one I didn't get very much at all. I wonder what this thing is. Oh, here's a crab too. There's no mistaking this one as a crab. This thing's like a crab that's almost ready to settle out. It's a late stage. So that one's about to settle out onto the bottom and be an actual crab. And I have no idea which one that is. Lots of crab species. See if I can clean it off. Get a better look at it, maybe. it may have lost some of its legs, which tends to happen in plankton collections. You know, these little organisms have been in a net, dragged behind a boat for minutes, hours sometimes, right? So they don't always come up in very good condition, but that's definitely a crab. It's about to be out of the plankton. As opposed to one like this, that's just like still a little crab, Crabzoia. And then we have our tiny mola mola. I shouldn't say mola mola. We have our tiny molid. I don't know if this is a mola mola or if it's another genus of molidae. We'll have to look at it in detail later on. Here's another one. Samples loaded with them. Every time we have something like this where there's a sample that's loaded down with them, I always ask myself, what can we do with these? What can we learn from all these fish larvae? We've got, at this point, in this sample here that we pulled out, we've got hundreds of them in here. So we might wind up with thousands of molid larvae in this sample before it's all said and done. And I always ask myself, cool, what can we do with them now? I don't know. I don't have an answer for that right now, but probably nothing. Probably, again, just get them cataloged and get them into the collection so that when in the future somebody does have an interesting question and needs some mola mola to answer one of their studies, you know, we've got them. They're available for research. I don't necessarily need to do a big research project myself. Just providing the material for others that's part of the job, right? That's part of the goal here. These are a resource, but they need to be available. These samples are in 70% ethanol, which tends to make this gelatinous stuff that's stuck to these things even like stickier. Dehydrates it a little bit and makes it even stickier. So the larvae, if they are stuck, they tend to get really stuck. And with all these spines on these molas, they tend to be very stuck. Getting them out of this stuff is tricky. But worthwhile and necessary. So you can see the alcohol. So again, I mentioned this is 70% ethanol. You can see that under this light that the alcohol is warming up a little bit and it's creating currents in the alcohol. And that's why you're seeing particles move across the field of view right now. We've created circulation within our alcohol. if you were wondering what all that was about. The alcohol's heating up and circulating within the dish. It's not heating up that much. Just enough to create some density difference. And you get circulation, even in a petri dish. What else we got in here? So many stomatopods. Pea crabs? Sand crab? I don't know what kind of crabs those are. All right. Let's go get more. You want to hop out of my chair? Daisy? Thank you. Every time I get up, the kitty cat finds it. Finds my chair. Takes it from me. Alright. So, yeah, back when we were doing the consulting world, you know, there's always going to be some loss of samples, or you're going to miss stuff. Not maybe loss, but maybe loss isn't the right word, but you're going to miss stuff. And so we had a rule that we were allowed, we had to recover like 90% of a sample. Science, we are definitely doing a science B. Finally, you know, Pad? Or I'm going to post this VOD up onto YouTube so you can watch the whole thing and take all the notes you want. This is a mola. Can it start from the beginning? Just watch, catch it on YouTube. So when I was doing the science consulting stuff, we had to recover 90% of the material. And so we would do QA, QC, quality assurance, quality control, on samples after we had finished processing them to we'd pull previously processed samples off the shelf and process them again and see if we were within that 90% allowance. So we might have been pretty good in the end. You know, the first couple of times you fail because you see things that are fish larvae you don't think are fish larvae necessarily. Some have some very weird forms. So I know that I'm gonna miss stuff doing this, but as long as I get 90% of the material out, then I feel pretty confident that from like an ecological perspective, we're gonna see whatever trends there are in the population, and we're gonna be able to the conclusions that we draw from those samples are gonna be valid conclusions, basically, right? So we have a lot of some sort of little crab larvae, right? Some little crab thing. That's what you look like in the morning? Yeah. Yeah. That's how I feel in the morning. Yeah, so crabs larvae go through multiple stages of this. You know, crabs are that's sort of the external shell, and as they grow, they have to shed their shell to get more space to grow, right? And so in the larval stages, when they're growing really fast, they shed these shells very quickly. There's high turnover, and every time they do it, they have a slightly different form. So to know crab, larval crab taxonomy, you have to learn 9 different stages of morphology. Zoea stage 1, Zoea stage 2, Zoea stage 3, like that. So it's ridiculous. Boil it and eat it eventually. I don't know what kind of crab this is. This might not even grow into a very big crab. So here's another. So this is another crab zoea, but just a much earlier stage than this one, right? Are these the same? I don't know. Probably not. Maybe. I don't know. I doubt it. Who knows? Who knows? Again, fish, not fish. So I don't know what these things are. Ultimately, we're gonna hand these off to our invertebrate. We have a crustacea collection, so we're gonna hand this over to the curator and the collections managers over in the crustacea department. They can figure out what to do with these things. Not alive. No, these samples were collected off of Hawaii in 1971, and they have been in storage, unprocessed for 50 years. So that's part of the reason that I really want to do this, is because, we have these samples sitting around where somebody took the time to go out and collect them, and then they're just sitting there. There's basically no value in having them just sit there. here's a fun one. Get this one queued up for you. It's huge. Looks like a leaf. That big thing. All those legs. It's kind of broken. Some of the legs are broken off. Hi, Daisy. They're eye stalks. This is the head. There's supposed to be eye stalks here that are broken off. But we've seen the stomatopod larvae, not the snapping shrimp, the mantis shrimp larvae. This is a phylosoma. This is a lobster larvae, and they get really, really big. So, you know, even under the microscope, that one's hard to see. But I'll take it out and see if we can see it under the. Like, that's a thing that you can see. That's a lobster larvae. What stage is this? Oh, brother, I have no idea. I have no idea. Again, I spent 20 years learning the fish. I didn't spend any time learning the invertebrates. Yeah, when I was working in Saudi, I had 2 technicians working in my lab who knew all the invertebrates. So I handled, like, my responsibility was to do the larval taxonomy, the larval fish. Sorry, try that again. My responsibility was to do the larval fish taxonomy and to do all the data analysis and report writing. And I had 2 technicians who worked in the lab full time, just processing the samples to get the data on all the invertebrate communities. So, you know, you got this sample is loaded down with shrimps. Like, whatever this shrimp is, there's approximately 1 gazillion of them in this stage or in this sample, and I have no idea what they are. Here's a fun one. I've been waiting for one of these. I assumed that there would be one of these in these samples, but we hadn't seen one yet. Squid. Anybody a fan of Squid Game? There's one now. Little baby squid. Squids are mollusks. Oh yeah, fish fact. That's what that's this whole stream, buddy. So squids are mollusks. So yeah, they have a shell, but the shell's inside the body cavity, inside the mantle. It's a little squid. Well, these are like porcelain crabs, I think. I forget what those crabs are. Fish fact. Well, hopefully we can find you a fish and I can give you a cool, like an actual fish fact about the fish that we're seeing. This sample is loaded with these shrimp and some other stuff. Those stomatopods, but it's just like shrimp. It's a shrimp sample. Shrimp. Shrimp this, shrimp that. There's a fish. That's what we're after. It's different about a mollusk from mammals, crustacean, or fish, or anything else. So they're invertebrates, right? So not vertebrates. no backbone. And what makes a mollusk? Oh man, it's been a you're really grilling me on things I don't know about here. Yeah. I don't know. I don't know what makes a mollusk as opposed to. I mean, they're like Mollusca is a huge. Is that an order? Phylum? I forget. Throw me a softball. How many oceans are there? 7 seas. Well, it depends on how you treat the North Pacific and North Atlantic, or South Pacific and South Atlantic. God. North Pacific, North Atlantic, South Pacific, North Atlantic, Indian Ocean, Antarctic Ocean, Arctic Ocean. I don't know, there's 7, whatever. There are no softballs. Your softballs are hard. That's not where my brain is at. So this is like a snapping shrimp. And so this one's like almost ready to settle out. I don't know where these samples were collected, but they must have been near a reef somewhere. Okay, even easier. What did you have for lunch? I had a ham and pastrami sandwich before I came on stream. So this is like a little shrimp. But I think this is one of those shrimps that burrows in the sediment, in the sand. Little claw right there. Different one. So I don't know if these are the same or if they're different. Again, shrimp. We haven't seen too many larval fishes in these samples. A lot of these mollas. And then relatively little else. Kind of fish see in future streams. So there's still a lot of this sample to process. So I'm going to, the whole point is to try to process all of these plankton samples that we have in the lab. So I'll be doing a bunch of these, but there are the samples that we have done so far have all been like very different community-wise. So this one seems to be monotypic. It's just a couple of things in here, but some of the other ones have had a bunch of stuff. And we have found a couple of things tonight. So far, we've seen a bunch of the molidae. There was a flatfish. So we got a bothid. A piece of a. It's a piece of a mantis shrimp. We got a Gampylidae, which is called a snake mackerel. So we've gotten a couple of things. Is this for fun or work? Yes. It's for fun because I enjoy doing it. And I thought it would be fun to do some of this on stream. But the samples are coming from work, and I am, like, the samples, once I get done processing them, are going to go back in for work. So all the fish that we pull out are going to go into the museum collections and be made available for scientific research. I'll get them cataloged and get them, properly in our database, and then they'll be made available to the broader scientific community. I don't have a specific project in mind for any of this stuff right now. Like, I'm not doing this to say, oh, I'm not looking for a certain fish to try to do a study on them. I'm just trying to get all the fish out. You should receive credit. No, it's just going to go into the collection. So there's nothing to give credit to at this point. Nothing to give credit to at this point. Although I've got some ideas. If this does well, I really want to show people more of like the scientific process. So I have thought about what types of projects we might do that would actually contribute to a proper publication, like a in a scientific journal, peer-reviewed science that we could do as a community. Those are like grand plans that are still far out there in the future. And here's another one of these funky crabs. I mean, part of it is going to show you guys like just how much time it takes to do these things. Do you know what I mean? Okay. Little crab. So it takes so much time. Like, it's crazy to me how we'll do a thing and it turns into a publication that's, you know, a six-page publication in a peer-reviewed science journal. Yeah. That represents, you know, 1,000 hours of work or something stupid. It's like so much work. And the return is like, sometimes it can feel so small, but oh well. That's the way it is. There's no easy way to do science, right? You just gotta sit down and go through it and do it. Alright. At least the kitty cat didn't steal my chair that time. Chair thief, Daisy. Okay. So much stuff. There's a huge pile of molidae embedded. Yeah, I got too much stuff in this one, I think. All right. Yeah, this sample has a load of these little dudes in here. Oceansunfish. And all different sizes too. So I don't know if these are all spawned at the same time or not. We really need to know if these were all spawned at the same time, because then that means there's a lot of variation in the growth rates between the different larvae. So if we just put these two side by side, there's not a lot of difference between the size, but there's a little bit. So if those were spawned at the same time, then one of them has had like a faster growth rate than the other one, right? But it'd be really hard to know if they were spawned on the same day. There is a way to age larval fishes. There's a structure in the inner ear of fish called an otolith. It's a little thing of calcium carbonate, and it's composed of a calcium carbonate and then a protein called otolin. And then in fast growth rate periods, the fish puts down more otolin into this matrix. And then slow growth periods, it puts down more, like proportionally, more calcium carbonate. And so what happens is, in fast growth, you can see through the otolin a little bit better. It's more transparent. And then in the slow growth, it's more calcium carbonate, which is more opaque. So you get these bands of light and dark growth periods. So we use otoliths to age fish on an annual basis. Like you can tell, summer season versus winter season. Oh my god, this thing is so entangled. You can tell like summer season versus winter season, right? Fast growth period when it's warm and food is available versus the cool season when temperatures are lower and growth rate's going to slow down. But it also works on a larval basis, on a daily basis. So when they're feeding, when they're active during the day and feeding, versus at night when they're not feeding. And so those are like the slow growth and the fast growth periods. So you can take out the otoliths of larval fishes and look at those bands and get daily growth estimates. You can tell how old a fish is. So in theory, in the ear of this larvae, there's a structure that we could pull out and look at it and count the rings in that structure and tell how old this fish is. In theory. In practice with these fish, it's very, very hard to do because this is a mola. This thing is so small that even extracting the structure out of the fish skull is going to be like virtually impossible. And then trying to get an age estimate from it is virtually like even more impossible. So in theory we could do it, but I think in practice it's such a hard thing to do, nobody's really even tried. Anyway, when I look at the different sizes of these things and I ask a question like, oh, are these things growing at the same rate? Were they spawned at the same time? Or are there different growth rates happening here? There's actually a way to test that. And look at that dude. Such a cool fish, right? To think that this thing grows into a almost 2,000 pound fish, wild. You can see all the spines that are sticking off of it, and yes, scientists like to name things, so all those spines on there have, like, names based on their position. Okay, all that junk off of this one. Snuff. Crab larvae, shrimps, mantis shrimp, which are not true shrimps, they're a different type of crustacean. So this is like the little claw of the mantis shrimp right here. Little thing with a big spike on the end. So that little thing. So that's the mantis shrimp claw. And they use that. It's like curled up, and they use that to like lash out and stab prey. Or if not stab them, they hit prey and will incapacitate them so that they can feed on them. And they're very, very strong. The adults are very, very strong. They've actually studied the musculature associated with those claws, because they can shoot those things out. It's some of like the fastest fast twitch muscles in the animal kingdom is in mantis shrimp. They can move the claws so fast that they cause cavitation underwater. If you're not sure what that means they can move so fast that they displace all the water and they create an air bubble. think about how fast they have to move underwater to move all the water molecules out of place and create an air pocket. That's what these mantis shrimps are able to do when they strike with that claw. They've been known to break out of aquarium tanks. They will smash the glass, crack the glass in the aquarium that they're in, or acrylic, I guess. Hardcore critters is what I'm saying. Pile right there. She's totally buried in something. These samples are in 70% ethanol right now, which means that things like, we're not seeing too many invertebrates, or not invertebrates, but too many gelatinous zooplankton. Because the gelatinous zooplankton at 70% ethanol, they rapidly dehydrate and they turn into just these gooey gobs. And so some of these fish, like the one I'm dealing with now, has some sort of gelatinous zooplankton thing wrapped around it. And now that it's dehydrated, like, all that stuff is just like glue. Kind of acting like glue on the fish. Yeah, nothing but molas in this sample. I mean, I say that like it's a bad thing, but we haven't seen a ton of diversity of fishes in this sample. It's mostly been the one thing. There's another one there. Cool, cool. Oh, that's right. We did get an eel larvae earlier.
With Asana, AI teammates can help level up your goals by connecting them to relevant projects, so every team knows their role.
Okay. I don't see much more than shrimp in this one left. Some Keating gnats, I think, yeah. So this sample is actually kind of rare in that we don't always get molidae larvae, but what tends to happen is the case that we have now, where they appear and when they appear, there are loads of them. You don't see them very often, and then you see just hundreds of them or thousands of them in a sample. So, you know, even though we are kind of seeing the same thing over and over and over again with all these molid larvae, that's still really cool because we don't have a lot of them in the collections, right? Hey, oh, finally another fish. Yay! A different type of fish. And a molid. Let's deal with the molid first and let's see if we can figure out what this other fish is. Again, it's got a bunch of stuff stuck to it. It's fine. Alright, what is this guy? Not the shrimp, the fish. So when we're dealing with most fish larvae, the mola is an exception because when you see a molidae, it's small, it's round, it's got spikes everywhere. It's pretty clear that that is molidae. There's not that much else it could be, right? But when we look at other fishes, like general fishes, there's a lot of things that we look for. General body shape is the first thing. So this one is elongate. It's long. Right? That's something that we pay attention to. We look at the size of the gut. So you see the mouth there. The gut actually is pretty short in this specimen. It only goes to about right here. That's like less than a quarter of the length of the fish. Yeah. We look at how deep it is, the body depth, and the body sort of thickness, how stocky it is. So this is kind of longer and skinnier. We pay attention to pigment. So there's a lot of pigment here on the surface of the gut of this fish. Pay attention to, again, pigment. So there's some pigment on the head here. And this is actually pigment that's literally pigment spots on the brain of this fish. And then we look at the fins. So the dorsal fin and the anal fins. We want to know where they start, where they end, and the relative positions of those fins. So looking at this fish right now, let's see, I need to flip it around so that we can get a good, proper orientation. So the dorsal fin is here on the dorsal side. The anal fin is here on the ventral side. And so we pay attention to things like, oh, the number of fin rays. It's hard to see that here. But oftentimes, the positioning of where the dorsal fin starts versus like where the anal fin starts, like if you draw a line through that point, you'll find that sometimes the dorsal fin is anterior, it's closer to the head, or the dorsal fin is posterior, it's closer to the tail. And those sort of relative positions can matter. It can help you figure out what family of fish that you're dealing with. Another thing that we pay pretty close attention to is myomere counts. So myomeres are muscle bands that are associated with vertebrae. So each vertebrae has a block of muscle that surrounds it, and that helps the fish move. So they're basically, there's a whole series of these things along with all the vertebrae, and so they can pull on those muscles and the vertebrae at different times, and that's how they're able to swim. You know, they make the S shape swimming. That's how they're doing that, is by pulling on the muscles associated with the vertebrae at different times. Coordinate that and you can make an S move. So we count those. And so we can actually see them a little bit here. On this, you can see there's 1, 2, 3. They're packed pretty close together. 1, 2, 3, 4, 5, 6, 7, 8, 9. So it's hard to count the vertebrae, but we can actually see the myomeres, those muscle bands that embrace the vertebrae, a lot easier. And since we know that the myomere embraces the vertebrae, there's got to be a vertebrae under there. We just count the muscle bands, and that is equivalent to the number of vertebrae that the fish has. So the number of vertebrae is an important factor in helping us figure out what family of fish this is. So when we look at fish ID guides, it talks about that sort of stuff all the time. Show you real fast. So I brought home a whole library. Early stages of Atlantic fishes. Volume 1. There's 2 volumes of this. This is like, there's 2,200 pages, 2,500 pages, something like that in this 2-volume set. And when we look at it, it has illustrations of the larvae, and it has information about all those counts, fin ray counts. So pull this up. I know it's hard to see, but One page has all the meristic counts, all the counts of things, and the other page has all of the information on like illustrations so we can see it, right? And so larval fish taxonomy really is about picture booking. It's about learning all the different forms of the fishes and knowing the resources to be able to go sort of key them out. I think that we're looking at Antherinidae. So again, this is something that we would do in a lot more detail in like once we have this thing sorted. But I just wanted to check on this one real quick and see if we could figure it out maybe. I think this is going to be in family 2 or in volume 2. I think these are Antherinidae. Again, based on the gut length, based on the pigment pattern, based on the myomere counts. So really what I want to do is see what the myomere counts are for this fish, and if it's in the ballpark, I know that's going to be what this is. So Antherinidae. 873. Yep, so that's what this thing is. This is an Antherinidae. So they have this long, skinny body, yeah, that I mentioned. It's elongate, it's skinny. They have a relatively short gut that's really compact up against the head. They have a little bit of pigment on the body, like it's mostly confined to the head. There's some on the body, but not that much. The mouth is also, it's slightly upturned. So we pay attention to like, is the mouth pointed straight out? Is it pointed a little bit up? Is it pointed a little bit down? The orientation of the mouth is important. And then the kicker for me is the total vertebrae count. These fish have between 37 and 40 vertebrae. And I don't know if it's picking up on stream very well, but I can see it a little bit here on my screen. But counting the vertebrae along the backbone here, there's like 30 something vertebrae. So I'm pretty confident then that this is an Antherinidae, a silver side. Anyway, so that's how we would go about like keying out and identifying larval fishes, and we would just do that all day long. After we get things sorted, that's kind of the process. Look for those key features, and then go through the literature to see if we can find something that matches those key features. Just like shrimp heads, crab larvae. Shrimps on top of shrimps. Yeah, and there's different species of crabs and shrimps in here. And there's so many, I just don't know. I never spent the time to learn the taxonomy of them. So grand vision here of what we're doing. Part of the reason that we're pulling out all these fish larvae is because we want to do... I want to host a larval fish taxonomy workshop where we go through and look at examples of things, and I can sort of teach people how to do this. So this is a ketognath. You can see there's the head with those chitinous teeth. And then the body's just a sac. But sometimes you'll find copepods in there or crab larvae in there. Not very discriminating about what they will feed on. Crab head. Or shrimp head. More shrimps. All right. Kind of moving through this one now. Yeah, just seeing like shrimps and stomatopods left in this sample. I think we've covered all the fish. That's gonna do it for that sample. Alrighty. So I think that's actually gonna... I think we're gonna start wrapping things up here now. But before we go, I just wanted to chat with you guys real fast. How do you feel about this stream? What do I need to do to make improvements? I've only started doing this today, right? This is the first time we're doing this, so... I think the plan is to do this primarily on Saturdays, is what we're going to do. I think this is going to be like that schedule going forward. Primarily do this on Saturdays, kind of in the morning, and then on Sundays I'm going to essentially like rip the portions of it out and put them up on YouTube. So I want to get some of the like descriptions and show people what a molidae larvae looks like, show people what the ichthyoplankton larvae looks like, and kind of go over some of those things and put that stuff on YouTube. I think that's going to be mostly what I'll be doing on Sundays. Saturday is going to be the main day to do the zooplankton sorting streams. Once we get a sample sorted, then we'll then go through and work on identifying all the things that we pulled out. This sample is going to be pretty easy because even though there's a lot of stuff that we're pulling out, it's all basically molidae and then a couple of leptocephalus larvae and some bothidae, and like we've got like 5 things that we pulled out today. That's all that we found. Not a lot of diversity. So yeah, I think that's kind of what we want to do. I just wanted to know if like how we doing. Does that sound like a good idea? Yeah, we're gonna get, you know, sort of my normal schedule is to play video games on Monday through Friday. So yeah, we've been doing video games Monday through Friday. So tomorrow, I think we're gonna do some more of Elder Scrolls Online. I'm expecting the Elder Scrolls Oblivion remake to hit sometime this week, so I don't want to start a brand new game until we know for sure if that's coming out. And I'm also expecting to get involved in the Marathon Alpha. So I think we're just going to do some Elder Scrolls Online until either one of those things comes into focus. On deck is going to be Bioshock, the Bioshock remaster. I've got Bioshock, Bioshock 2, and Bioshock Infinite ready to go. So we'll be playing those during the week. And then on the weekends, we'll spend time sorting plankton. I think that's going to be the goal and the plan moving forward. I'm going to make a couple of tweaks here to the setup. I think I need to change a few things around. I want to make I'm going to get a little table that I can set set over here by me so that I don't have to get up every time we finish processing a dish. I don't have to get up and run over to the kitchen to swap things out. It's a little bit of an annoyance. So we're going to try to make it easier to do that and move things faster where I don't have to like get up and walk away. So I've got a few little changes in mind, some setup stuff that I want to try differently. But otherwise, yeah, I think this kind of worked. I had fun doing it today. I hope that you all hopefully had fun and learned some stuff. And yeah, appreciate you all being here. And we'll do it again next Saturday is the plan. Tomorrow, ESO. Saturday, more plankton. All right. So thanks everybody for watching. Thanks for hanging out. Appreciate you all being here. Thanks for all the follows we picked up today. And I will see you all next time. Bye.